get started. Sorry for the delay there, and thank you for braving this cold weather and surviving so far. This is probably one of the worst winters I've remembered. So the snow blowing has been endless. So I would now take an opportunity to introduce Dylan Cable. He's an assistant professor of biostatistics and earned his Ph.D. in electrical engineering and computer science at MIT. He's done internships from coast to coast, as I noticed, and had multiple publications, typically starting with the word nature in the journal or science. So very high level stuff this guy's been doing. Has had multiple awards, but the one that was interesting to me was that he was an MIT arts scholar. So I'd like to know a little bit about that. Thank you. And Dylan will now be presenting.
Great. Well, thank you, Evan, for the invitation and the introduction. Can everyone hear me?
We can't see your presentation.
Oh, we can't see it. Okay.
There you just keep moving. Hello, Vanessa.
Cut off.
Okay. Think it should be good now?
Yep.
Okay, great. So yeah, today I'm going to be doing an overview of the research that our group has been working on, mostly in spatial transcriptomics, but also some other areas. And then I'll be going through a few different projects today, so please feel free to interrupt me at any time with some questions. Okay, so this is just an overview of our research program. So what we do here at U of M is we develop a combination of statistical machine learning methods for emerging high throughput technologies. And this includes spatial transcriptomics and single cell RNA sequencing, and then also perturb sequencing. And then a newer area for us is in some biological sequence engineering. So if that's protein or RNA sequences. Today I'm going to be focusing mostly on our spatial transcriptomics projects. So I'm going to go through some prerequisites for doing spatial transcriptomics analysis, which typically the first step is assigning cell types. So that'll be the first. And then once we do that, then we'll go on to how do we determine patterns of cell type specific spatial differential expression. And then I'll go to some more recent projects we have in spatial allele specific expression, as well as unsupervised cell type identification. Okay, so I'm sure most of you are aware of this. This is an overview conceptually of what is spatial transcriptomics. So for those who are aware of single cell RNA sequencing, we typically measure each cell, which is shown as a dot, in gene expression dimension. So this is a schematic of expression dimension 1 and expression dimension 2. And what we can do is we can color the cells by cell types. And in single cell, we're pretty fortunate because they tend to organize into these distinct clusters. And you can use clustering algorithms to assign identity to each of the cell types. So this is in the cerebellum, part of the brain. And we have 3 cell types shown here: Purkinje, Bergman, and granule. And then spatial transcriptomics is similar to single cell, but now what we do is we actually have, in this case, for this technology, we have an array of beads that captures the spatial location of each cell. And of course, there's multiple technologies out there, but what we get at the end of the day is this input matrix, which is called digital gene expression matrix, which is number of pixels or spots by the number of genes. And importantly, we don't assume that each individual location is a particular cell. So, and we also know what's not shown here is the spatial location of each of these spots. So some of the questions we're going to go through today is how can we detect patterns of gene expression across tissue, and how can we account for mixtures as well as technological effects in these data sets. So the first problem that we're going to focus on today is how do we identify cell types in spatial transcriptomics? And so here is a schematic of one of these spatial transcriptomics sections where it's colored by cell type. And note that I've set it up so that each pixel can actually be a mixture of multiple cell types. For example, these ones in the middle are a mixture of Purkinje and Bergman. It's referred to as the Purkinje-Bergman layer of the brain. And the goal here is to create a map to identify the identity of each cell type. So what this is that each pixel is going to be mapped to some proportions, where here what we're going to do is to infer the proportion of each cell type on each pixel, where those proportions are going to add up to 1. And then here we have an overview of some of the different spatial transcriptomics technologies. So we have ranging from subcellular resolution. These tend to be imaging-based spatial transcriptomics platforms, such as MERFISH. And you can resolve one cell type through segmentation, although that process is not perfect. But we also have to be aware of that some of these other technologies, such as Biostatistics and Visium, which tend to be capture-based, for those technologies, they can range from 10 to 100 micron resolution, and you tend to get 1 or 2 or even more, 3 or 4 cell types per cell. So we're actually going to keep those different cases into account when we go through this. So this is our team here at U of M. We're made up of mostly Ph.D. student, master's students, and undergraduate students. And this is our team that works on spatial transcriptomics. So the first thing I want to point to is why is this necessary? So as a motivation for why you want to consider cell types in spatial transcriptomics, one of the first things you might want to say is, what if I don't care about cell types? I just want to find genes with interesting patterns. Well, this is one of the first things that people tried with spatial transcriptomics. Some popular metrics are ones such as Moran's eye to find spatial patterns. And if you just naively apply this to spatial transcriptomics, so this is the hippocampus within the brain, these kinds of genes come up. So this gene is called PTK2B, and it does have a spatial pattern. So the way to read this plot on the left is that the bold represents the detection of the gene, and then the color, we also superimpose that with cell types, where blue is neuron and red is all other cell types. And what we can see here is that this gene definitely has a spatial pattern if we didn't know anything about cell types. But what we can also see is that this spatial pattern is entirely determined by the pattern of the excitatory neurons. So we call this a marker gene. This is clearly a marker gene for excitatory neurons. And I would argue that this is actually not the most interesting use of spatial transcriptomics, because we already know the marker genes from single cell data. So we could have just determined that we know that the excitatory neurons follow this pattern. And then what's actually more interesting is if we control for that and look for patterns within cell types. So that's kind of why cell types we view as a confounding variable. And first step is to determine them and control for them. So you can see this pattern more generally here, where this is quantified as the coefficient of variation across cell types. So we looked at spatially variable genes that are detected using this method, and they all have a lot of coefficient of variation across cell types, indicating that they are marker genes. On the other hand, we compare this to randomly selected genes, which have a much lower average. Now, what we propose is that we instead take one cell type at a time, for example, the CA3 neuron cell type, and we look for genes with spatial patterns within that cell type. So we remove cell type as a confounding variable. And this is the original set of genes, and this is the coefficient of variation within CA3. What you can see is that the genes, such as this one, actually don't vary that much within cell type. But if we specifically look for genes, then we can find a bunch of genes that do have a strong variation within cell type. So hopefully that's a bit of motivation. So this is a setup of the problem here, where we're going to take into account spatial transcriptomics. And today we're going to be talking about both unsupervised and supervised methods for cell type assignment. So what do I mean by supervised? I mean in a machine learning sense that we can be training on one data set and then testing on another data set. And the data set that we're going to be learning from is the single cell RNA-seq data set. And the thing is that later in the talk, we're also going to consider unsupervised methods where we discover the cell types directly from the spatial transcriptomics data set itself. But anyways, the data that you can think of here is that each spot or pixel on the spatial transcriptomics has some true underlying composition of cell types. In this case, it has some proportion of red and some proportion of blue. And we're going to be thinking about here the data generating process that's generating this is that we think that each gene is contributed by each cell type in accordance to that cell type's tendencies. So for example, this gene 2 could be a marker gene for the red cell type, and that would come entirely from the red cell type. Whereas gene 1 might be more of a mix and come from both of them, and so on. But we don't actually observe this, so this is kind of the generative model. But what we observe is black box, where we just get the total counts and we don't know the identity of the cell types. Okay. Some final motivation that I want for some of the challenges is I've mentioned this problem of cell type mixtures in the slide-seq cerebellum. And now I want to show you why this exists. So this is the Bergman-Purkinje layer. And on the left, I have a spatial plot. And on the right, I have a gene expression plot. And these two cell types, you can see that they're overlapping physically within the same layer. And then on the right, when we plot them in gene expression space, we can see that although if this was true single cell, you would see an L-shaped with two groups at the edges. We plot marker genes on each axis, and you would see no overlap in the middle. But what we actually see in this case is we do see a population in the middle, which indicates mixtures. And here we see what happens if we naively apply standard single cell clustering to this spatial transcriptomics data set. Then the color is shown in red or blue, and we can see that clustering is unable to identify these center ones as mixtures and instead creates arbitrary assignments between two cell types. So we're going to try and improve on this. So that's one challenge, is the cell type mixture. Second challenge that we're going to deal with today is that in a supervised method, when we're training on one data set and testing on another data set, we get very different effects of those two data sets. So here, this is showing within platform performance of a very simple classification method, which is linear regression. So if we use linear regression to classify the cell type and we train within, this is single nucleus RNA-seq, then you can read this as a confusion matrix. So this is for the true cell type, the proportion that it's predicted to be each predicted cell type. So perfect performance here would lie along this diagonal, which is highlighted in orange. And we see that it's not too far off. The biggest error comes from this fibroblast endothelial, which are similar cell types. But now what we do is we're going to take two similar data sets of the cerebellum. One is single cell and one is single nucleus. And here we're going to train on single nucleus and predict on single cell. And when we do this, we see that the accuracy goes way down. And we attribute this to technological differences between the two platforms. We know that there's different genes, such as nucleus-specific genes versus cytoplasmic genes. So it's actually, we do have to do a bit of a correction. We can't just naively do it. So that's an overview of the challenges. And now we're going to go into some of the methods for how we approach this problem. So this is our paper for robust cell type decomposition. And what we do here is we're going to fit a probabilistic generative model to do this supervised classification. So, and you can read more about our paper here. I want to go into some of the mathematics of how we do this. So to start, we're going to start with a standard Poisson sampling assumption, where here we have the gene counts at pixel I, and gene J is defined as Yij, and those are integer counts. And what we're going to assume is that this is drawn from a Poisson distribution determined with a mean by two things. One is the total number of counts, Ni, so we assume that we're scaled by Ni. And the second is the underlying gene expression rate, which is given by lambda ij, where again, I is pixel, j is gene, and those are going to be multiplied together. And what we see is that we can model this lambda ij is a linear combination of some weights for the proportion of each cell type. So this is beta ik times the underlying profile of that cell type, which is mu kj, where j is the gene. So what we're saying here is that we're assuming that each pixel is going to be a weighted average of the underlying pixels at that location. So if you have a 30%, 70% mixture, then each gene is given by 30%, 70%. Okay. And then the Poisson takes into account the sampling distribution, but we have an additional term for any additional biological variation, which is assumed to be log normally distributed. So now I want to talk about this platform effect. So how can we quantify the platform effect in our model? Well, like I said, we can actually measure the true platform effect if we plot within one cell type the gene expression of each gene from the single nucleus versus single cell. And this is a scatter plot here. And you can see that while these two platforms are correlated, there's a lot of variation, which is deviance from the XY line in these platforms. So we can quantify this by defining the platform effect to be the log ratio of gene expression. And we see that in log space, this is approximately normally distributed. And this motivates us to define this platform effect, gamma j, as a normal. And the gamma j is gene specific that's shared across the entire data set. So this is kind of how we use the data to motivate that we should actually be using the normal distribution here. Okay. So now I want to talk briefly about our estimation framework. So this is the RCTD model, and the way that we fit this, I'm just going to go through a little bit briefly, is that we first estimate the cell type means mu kj from the single cell reference. And then after that, we're going to predict and remove the platform effects, gamma j, using the pseudo bulk RNA-seq counts. And then finally, what we're going to do is we're going to estimate the cell type proportions, beta ij, independently for each pixel via maximum likelihood estimation. And now I'm going to skip over some of the math today, but essentially we frame this as Poisson log normal regression, and we create some second order Noonan-Raphson based optimization method, which reduces to a sequence of quadratic program approximation for the log likelihood. So I'm not going to go into too much of that today. And then the platform effect normalization procedure, the details are also shown here, but I'm also not going to go into this today. So I'm going to go to the results of the method. And it looks like we have a question. So thank you for the question on Zoom. So are there fewer platform effects if comparing single cell RNA-seq versus spatial instead of single cell RNA-seq for single nucleus RNA-seq versus spatial? Yeah, that's a good question. Thank you. So if we go back to our model, we can see that parameter of our model is sigma gamma, and that stands for the magnitude of the platform effects. So we do actually estimate this parameter, which tells us the size of the platform effect in our model. And the comment is exactly correct, that when we do measure this, we do tend to see that if we're doing it on spatial and we start with a single nucleus RNA-seq, then we introduce the most platform effects compared to using, for example, single cell RNA-seq. But of course, single nucleus RNA-seq is the most common, but we do think that the isolation of the nuclei is what introduces the most platform effects. And of course, there are some spatial transcriptomics technologies that are closer to single nucleus, but that wasn't what we tested it on. Okay, and then what's the reference for true cell types is another question. So the reference for true cell types, that comes from a single cell data set. So most single cell data sets are widely available. There's a lot of papers on single cell, and most of them include some kind of validated cell type annotation. So typically we're not trying to redo that analysis, but we do take that as our input for this algorithm. So we treat that as the truth, even though there could be some limitations of that data set labeling. Okay. So this is some simulation that we set up to validate how we're doing, where we're going to use the single nucleus and the single cell data set that I've been talking about to generate a simulated spatial transcriptomics data set, where what we do is we start with single nucleus, then we add platform effects just by using the single cell. And then what we're going to do is we're going to create simulated mixtures of single cells by adding these together.
Oh, I wish there were fewer.
And then that's how we generate our simulated spatial data set. So if we do this and then we run our algorithm, how does it perform? Well, the first step is that our platform effect estimation, we pretty much predict about 90% of the variance of these platform effects. And when we remove that, then what we see is that the cell type classification is much more highly accurate. And we think this is because of dealing with the platform effect.
Yeah.
The next issue that I want to discuss is that we look at this mixture issue, where before I mentioned that we have this potential mixtures of Purkinje and Bergman cells. And then what we're actually going to do here is we're going to classify them into, some of them will be labeled as single cell singlets, and then some of them will be labeled as predicted doublet or mixtures. And then what you can see is generally the marker composition of the marker genes reflects the relative composition of those two cell types. And furthermore, we do something else, which is that in the mixture case, we estimate the proportion of those, which is shown in color, the relative proportion. So all this here is, this is the output of our algorithm, where we can create these spatial maps of cell type predictions, where each cell type is labeled according to its color of the predicted cell type. And here we're able to reconstruct some known layers of the cerebellum on the left, and this is the hippocampus in the right. For example, here you have the Purkinje-Bergman layer in blue and red I mentioned, the granular layer in green, and then the oligodendrocyte astrocyte layer in the center. So I'm just going to pause there for any questions before moving on to the second part of the talk.
Can I have one question?
Yes.
So, great talk. Thanks. One question is that, the recent discovery, there are a lot of extracellular vesicles, also called EV. So how do you distinguish is a true cell, true single cell, or is just the extracellular vesicles instead of cells?
Extracellular vesicles. That's interesting. So most of these are RNA based rather than protein based. And I'm just not familiar with this. Do extracellular vesicles typically carry RNA?
Yeah, they carry RNA, protein,
Okay.
or membrane. So they have almost like the cytoplasm component out in the extracellular vesicles and secrete outside of the cells.
Yeah, so that's a good question. So is that something we've explicitly considered? I will say, so this algorithm doesn't distinguish between the source of RNA, but if you do have multiple components, then that can be dealt with as a mixture. So I can say an analogous thing that happens, which is that in the brain here, we have this strong layer of neurons, and we have the cell body, the soma, and we also have the axon and dendrite. And what you do see is that if you measure the RNA in this tissue, you do get the highest concentration of neural RNA will be found at the soma of these cell types. But what you do see is some smaller proportion in the extended neuron and in the extended axon and dendrite region. So you can kind of think of that as an analogy to the extracellular vesicles, or what happens if you have this distributed low amount of RNA. What we see is we typically don't classify those as individual cells because we don't detect enough neural RNA at those particular locations. But I think that it would require further algorithm development to explicitly properly deal with that case, but we don't currently necessarily think particularly hard about that issue. So I do think it's an interesting area of research. Thank you.
Thank you.
Okay. Another question. Okay, so when discussing singlets and doublets, doublet can include more than 2 cell types. Yeah, so we can classify it as a doublet. In this example, doublet was exactly 2 cell types, but you can analogously have more than 2 cell types. Have you tried ground truthing using histological staining? So we haven't actually tried that, but that would be interesting to try, but we just haven't tried that yet.
Jocelyn Benson, in February 17, 18,
So
not February, but March.
okay, if a spot is a doublet, which cell type do you assign to the spot? So that the answer would be both. We think that two cell types can be there simultaneously. Okay. Well, thank you for all the questions. I'm going to continue to the next part of the talk, and then happy to answer more questions there. So now that we talked about how to identify cell types in spatial transcriptomics, we're going to talk about inference of cell type specific differential expression in spatial transcriptomics. So here to do this, what we're going to do is look at this particular example of differential expression, where what we have is here are these 2 cell types, Purkinje and Bergman, that do appear to be in the same layer. And here we have 2 regions of space that we're going to consider for differential expression. So this is the anterior region in green and the nodular region in red. And what do we mean by cell type specific differential expression? What we mean is that we know that genes can have a particular spatial pattern. So this is the ALDOC gene, which is shown here. And what we're going to do is that we're going to try to figure out which of these genes is responsible for the spatial change that we see between the anterior and the nodular region. And so to do this, we're going to use a technology here called HCR imaging, which is a very low throughput technology, which will give us cell type specific images of gene expression in this ALDOC gene. And what we get when we see this is we see a strong spatial effect in the Purkinje cell. However, for the Bergman cell, even though it also expresses this gene, we see a small spatial effect. So we want our algorithm to be able to replicate this fact that this gene has a spatial change in the Purkinje cell, but not the Bergman cell. So what do we mean by cell type specific spatial expression? Well, there's actually a bunch of different patterns that we can have. So these are some of the use cases of our algorithm, where here we can have the discrete region spatial expression, which I mentioned before, but we can also have a continuous distance, or we can have some smooth nonlinear spatial pattern. And then we can consider other cases such as cell-to-cell interaction, where we have the density of one cell type. We can also have proximity to some pathological element, such as we've applied this to plaque in the Alzheimer's disease, or we can consider the cellular microenvironment, which is defined by the local neighborhoods of the cells. So here, all these are going to be treated as covariates or predictors of our model for spatial gene expression. And I just want to emphasize this point, is that a lot of these problems have been treated as their own problems, such as cell-cell interaction or tissue microenvironment. But here we can actually unify these in one framework for spatial differential expression, which we believe is the cleaner approach for doing this. So this is an overview of the seaside cell type specific inference of differential expression project, where we start with, again, the spatial transcriptomics. And here we're going to build off of our earlier RCTD algorithm for assigning a spatial map of cell types. And here we're going to additionally take into account some user-defined covariate of biological interest. So here we've defined this covariate to have two spatial regions, the yellow region and the pink region. And then we also consider the individual gene expression, in this case called gene X, that we're looking at between the two regions. And here you can see the cell types as defined by before as addition to this white circle, which represents expression of gene X. And how this algorithm is going to work is that what it's going to do is it's going to estimate gene expression in each region for each cell type. So here you might see that within the green cell type, we get a higher level of gene expression in the pink region compared to the yellow region. But then the other cell types could have the opposite pattern. So that's why we call it cell type specific.
Yeah.
And after we have that, then we can do some tests for differential expression. Where here what we have is we color each gene within each cell type as significant if it comes up as significantly differentially expressed one way or the other. And more about our paper is here in this journal. So to do this, how are we going to do this? Well, we're going to start with our original model. And the main idea is that we're going to extend it so that we no longer assume that the gene expression of a cell type is fixed. So before we had the cell type profile was mu kj, and now we're adding an index I to account for the effective space. And how do we actually model these? Well, we assume that these mu's vary log linearly as a function of the covariance. So our covariate matrix is shown in X, and it's going to get multiplied by some coefficient of the effect of those covariates. So an example is if we have two regions, then we would use a dummy variable covariate, which has ones where that region is and zeros everywhere else. And it gets multiplied by a single coefficient alpha. And the size of the alpha, these are the parameters of our model, tells you the amount of spatial differential expression inferred by the algorithm.
Get in here.
So this is the overall model. And to implement this model, we just use a trust region-based optimization method, which I'm not going to go too much into. But I just want to go through here. This is our simulation for this model, where here we have a similar setup to before for the simulation. But here we have two spatial regions called 0 and 1, and we're actually going to change the gene expression within that region. And what we're going to do is we're testing cell type specific gene expression. So we're only going to change the differential expression in cell type A, but then we're going to see how does the algorithm perform in estimating the gene expression of cell type B. So here we compared to 5 different methods, including our method Seaside, which is in this red color. And the ground truth is shown in black. And the ground truth would be 0 DE, because we're not actually changing the differential expression of cell type B. But we do see that a few other methods can run into some failure modes. For example, the most commonly used method for differential expression is the bulk differential expression method. So the bulk DE method simply will pseudo bulk each of these regions and then compute the differential expression between the regions as a pseudo bulk. But it ignores the cell type. So what we find is that if we vary the difference of mean cell type proportion across the two regions, just changing the composition of cell types, that this bulk method will think that there's differential expression, even though those cell types themselves are not changing. So our method controls for that. But then what we see is that even if we keep the cell types fixed, but now we're just going to change the differential expression in one cell type, but not the other, then we see that many of these other methods actually get confounded by this. So one method I would like to point out is this decompose method, which is another set of popular methods where people want to take this composition and split it into single cells. So if you have a mixture, then can you predict both of the single cells? And the problem with that is if you have differential expression, you're just looking at one cell at a time, you don't actually know which cell type is responsible for that. So you just distribute that among both and you get some confounding. So we're able to handle these cases. So we also check that our method, when we do the test, has a good FDR control. This is shown here. So we have different significance levels and false positive rate is well controlled. And now I want to go into some biological validations. So what we do here is these are the two regions of the cerebellum. And what we're going to do is we're going to measure the DE using both our algorithm and also a low throughput technology, which I mentioned, called HCR. And HCR, we're able to only measure about 6 genes. But here the cell type is shown as the color. And we also have confidence intervals of our algorithm's predictions. And so what you can see is that most genes generally fall within the predicted range of our algorithm versus the truth that is measured. And while it's not perfect, I would say that this is some strong evidence that we are able to distinguish between cell type specific differential expression. For example, the ALDOC gene, we have that's predicted to be high level of DE in the red cell type, but not very much in the blue cell type. And that agrees with the ground truth here. So that was the ALDOC gene from before. And we also extend this to some population level DE, where if we have not just one spatial transcriptomic sample, but multiple, then we can integrate that to get a population level spatial DE estimate. And then we further extend this to some meta-regression framework, where this can take into account more complex spatial transcriptomics experiments, such as if you have case control, healthy versus disease. We have position of the sample spatially within some tissue or different time points of the tissue. So that's something else that we're working on. So that's it for the spatial differential expression. So I'll pause there for some more questions. Yes.
New questions. Maybe I missed it.
Yes.
When you compare the different locations in gene expression difference,
Yes.
do you have any normalization before?
Yeah, that's a good question. So to compare different regions, the normalization that we're using of the gene expression. So here we're using count data, and the normalization in our model is that I go to our model. Our data is given by yi, but we do also take into account the total number of counts ni at each location. So just because if you have more depth of sequence at that location, we would expect to get twice as many. So we assume that it's this proportional kind of normalization. But then the other kind of normalization is the reference that you're using to compare. So we can't just get an absolute change without having a baseline. So that's why we need 2 regions. You can't just say, is this gene differentially expressed in this one region? Because we need to know with respect to what's the baseline that we're comparing to. Great, any other questions here?
Dylan, I got
Yes.
one. So when you do the differential expression as you showed here,
Yeah.
you said that FDR is good within that tissue. So the question becomes, how many different biologic samples do you know to say that is actually statistically important?
For sure. Yeah, so that's a great question. So that's where we come into this multi-sample analysis, where we find that there's a very big difference. So most spatial transcriptomics analysis including for our original paper on this topic, was done on single samples at a time. And it's very easy because as we've seen, we have 10,000, 20,000 data points. We can learn a lot about this sample and say there's a lot of differential expression in this sample. But biologically, it's not actually very meaningful unless it generalizes to other samples. So what we find is that when we include other samples, for example, if we have some spatial transcriptomics data set with 4 replicates, 5 replicates, then now we're actually looking for a DE that replicates across the population. And we typically find much fewer hits in terms of significant, where you might have found 200, 500 genes on your original, but now you're only finding 20, 30 significant genes in the population. And that just comes down to sample size and statistics, where your limiting factor is not the amount of information you have on each sample, but it's actually the number of samples. So actually, if we can magically, for the same amount of money, get twice as many samples, but half as much data per sample with spatial transcriptomics as it currently is, we would probably make more discoveries on the population level. So one of the downsides of these technologies is that they're really expensive per sample. You actually get too much information, too much sequencing per sample. So then the flip side is you don't have enough samples as much as you would like ideally for population level inference. So this is a problem that we're actively working on here. And it gets more complex when those samples are not just the same, they're not just replicates, but they're actual test cases. So case versus control, you're adding in age covariates, whatever you want to test for in the spatial data, it gets more complex. Yes. Yes.
Maybe I didn't quite catch on. So in the example that you gave, you picked out the regions and you wanted to say, are they different between these regions? But if we had a sample and we wanted to know where are the regions of variability, like we have, for example, if you're near cancer cells, for example. So if you don't know the distribution of variability, how will you back that out?
Right. So our approach is pretty flexible, but it's also user or expert driven, where having input from the user will help to get better results. So let's say you don't just want a simple region case, but you actually want to look at cell-cell interactions. So a common question that we look at is tumor immune interactions for cancer cells to immune cells. So here what you would do is, for example, we could have the immune cells be the covariate, and this is a continuous covariate where we capture the density of those immune cells, and we look at how the variation of the cancer cells changes with respect to those immune cells. So I think from what you're saying, that would be a really interesting thing to do. If you actually can't figure out what you want to test for, you don't have any concrete hypothesis, then you can look for general areas of variability, which is smooth spatial patterns. So this is a non-parametric method that doesn't make any particular assumptions of where the variability lies, but discovers it. So you tend to get more discoveries if you come in with a hypothesis, but you can also use it without a scientific hypothesis. So hopefully that clears things up.
Thank you.
Yeah, I'm trying to see if I don't think I included the slides of the tumor immune cell case, but okay. So now I'm going to go into some of our other projects that are more recent, which is spatial allele specific expression. So this is a bit of a niche scenario that we've also applied this to, but you can actually also get allele specific or isoform information from these transcriptomic data set. So what you can is within each spot, not only do you get the gene identity, but you can also get the allele, which is here shown as the maternal allele in red or the paternal allele in blue. So what we have is this composition of both cell types and varying gene expression and also varying allele composition. And the question that we want to ask here is, within this data set, can we test or fit these similar models to get allele specific expression? And there's multiple different types that you'd want to consider. So one is global spatial allele specific expression, where you have this variation of allele composition, which is shown as the color going from red to blue and white being in the middle. And then you can also have within cell type allele specific expression, as shown here, where it's not actually spatial, but it just depends on cell types. And then finally, you can have within cell type spatial allele specific expression, where it's within cell type. So this is pretty much everything here is analogous to what we just did for differential expression, but now we're also doing it for allele. So even if the gene expression itself isn't changing, looking within that. And we can actually modify our original model and instead of using the Poisson log normal, we use the beta binomial distribution. So there's more details here in our paper. And then I'll also say that this is a little bit of the calculation that we do, but I'll just say that this actually builds upon the seaside algorithm, where now we need to know about the cell types and also the gene expression, spatial patterns. And then we can look at within a gene, either the alleles or the isoforms. Okay, and now I want to, for the last project I'll be focusing on today, talk about our work in unsupervised cell type identification. So here we're going to go back to the problem of cell types and then say what happens if we remove the reference and then just look at, can we identify the cell types directly from the spatial data set. So here I want to go back to some of the fundamental issues with this data. We're going to look at how feasible is it to do this platform effect estimation. Well, the thing that we can actually discover here is not only are there platform effects that we showed, but we can also get cell type specific platform effects. So this is going to be a little bit complicated. So I'm going to try and hopefully walk everybody through it. We're actually going to look at the log fold change between two cell types. So here we have the true log fold change between Bergman and Purkinje versus the predicted log fold change of Bergman and Purkinje. And this is going to be in two different platforms. And if the relative difference between two cell types stays the same, that's actually what's more important. So as I discussed before, we talk about global shifts in cell types. We can correct for that. But with our platform effects, we don't actually change for changes in marker genes. And so these two cell types are actually relatively well behaved, where in the green, you see that the change is well predicted between these two platforms. But what we actually get is within two more similar cell types, such as poly or oligodendrocytes, or fibroblast endothelial cells, is we see that there's actually much less correlation between the marker genes between those two cell types, or the DE genes, in one technology versus the other. And that actually violates one of the assumptions of RCTD, which is that our platform effects are changing across all the cell types at once, not the differences between the cell types. So we see that in both of these. And this actually is why we think that we have a lot of errors from the supervised algorithm. So that's the cell type specific platform effects. The other issue is that we talked about when you apply unsupervised clustering, and if we apply it to this other data set, we can actually get entire clusters that are mixture clusters. So this obviously is not good because we don't want to think that an astrocyte neuron mixture is actually a novel cell type, because it's not. So this leads us to consider some factor-based unsupervised method to discover cell types. So that's actually what we're going to develop here. And what is a factor-based method? Well, factor-based method is going to learn the cell type factors, and through some matrix factorization, it's also going to learn the relative weights of each of those factors on each of the locations. So there's this joint two-part learning problem. And here we're going to look at what happens if we just apply some standard non-negative matrix factorization. Well, if we apply non-negative matrix factorization, then let's look at the sparsity here. So here we're going to look at This is a data set, the spatial data set that we know is sparse. So we know that the truth, which is shown in black, is that each location will have either one, in this, on the case on the left, it'll have a proportion of 100% of the first cell type. So this is the order of the factor ranking. And then if we consider the doublet case, such as slide seq, where we think there's singlets and doublets, but nothing more, then again, we have the truth has this. But when we apply NMF, it basically spreads out the weights much more on the different factors. So we refer to this as the sparsity problem. The real spatial transcriptomics data is very sparse. It only has a few cell types per location. But if you discover factors, they're going to be more spread out. It might learn something like cell cycle or some shared neuron factor that's kind of everywhere, and it doesn't look for those specific sparse factors. So we can actually, and then you can also see this if you look at the factor themselves, how specific are they for cell types? All of them are below, they hover around 50 to 30% specific for individual cell types. So if we just apply an out-of-the-box factor method, it doesn't specifically discover cell types. So the way that we think we can get it to discover these is by imposing a sparsity constraint. So this is the project that in our group that we modify our original algorithm to be sparse and also to go without a reference. And we refer to this as reference-free inference of cell types and expression, or RICE. And this algorithm is an iterative algorithm that alternates between two tasks, which is that first, we assume that we actually know the expression matrix. Well, if we know the expression matrix, then now we're in the RCTD problem, and we can just estimate the cell type proportions. But what if we don't know that, and now we just know the cell type proportions? Well, if we know the cell type proportions, we can estimate the gene expression matrix, and that actually can be done through a modification of the seaside algorithm. So we actually can iterate back and forth between these two with some random initialization, and we repeat until we get convergence. So this is the math here. Again, it's similar to the RCTD model, but now we're actually going to assume that these mu's are unknown. They're not going to be calculated from the single cell reference. And if we do this, let's go back and look at these earlier ones of can we actually predict the log-fold change, the relative marker gene ratio between these two cell types. And we see that even for these two very similar cell types, we're doing a much better job of the supervised algorithm, which would have been shown in green. That's what RCTD would have been using. Now we're doing what the unsupervised method was shown in red. And again, the truth here would be the y equals x line. And how are we doing for sparsity? Well, our sparsity metrics are actually lining up pretty closely with the data. What this means is that if we apply it to singlets on the left, we are saying that those are pretty sparse with respect to the factors that we're learning. We apply it to doublets. We're again getting something pretty sparse, pretty similar to the data. And if we look at the identity of our factors, they're hovering more around 80 to 90% composition within one cell type. So our factors are actually learning the cell types. So this, how you interpret this is within the factor, what proportion loads onto a single cell type. And then the other thing that we can do is we introduce a pruning procedure to deal with issues like this. So we actually use clustering to initialize this algorithm in a lot of cases. So when we're starting off this algorithm, we can end up with clusters like this, which are actually just mixtures of neurons and astrocytes. And what we do is we prune the algorithm by saying, what if we remove this cell type or factor, and then we refit it, and we look at the change in log likelihood. And what we see is that if the change in log likelihood is below a certain threshold, which here is set to 25, then we actually prune, get rid of that cell type. It means the algorithm is just as well off without that cell type. And we can see here is that the two cell types that were pruned in this experiment were these ones in gray that were labeled as these mixtures of astrocytes and neurons. So that's a little bit of that. here's basically the output. This is a lineup of the marker genes, the rice predictions, and the RCTD predictions, and we can see that we're pretty much replicating RCTD's behavior on the cerebellum data set, but we're not using the reference. And so this is an overall comparison between the RCTD and the rice confusion matrix, and we see that there's a lot of agreement here. I want to conclude by just mentioning our R package, which all these methods we put on our SpaceXR GitHub, and then more recently, our package has been accepted to Bioconductor, and it is actually published on Bioconductor right now. So the version that we maintain is on Bioconductor. That includes RCTD, and then for the newer methods, we just put them on our GitHub before we migrate them to Bioconductor. So we have about, depending on the technology, hundreds to thousands of users on of our software. So that's pretty important for us as well. So that's it. I'm now just going to go to, whoops. It went to that. Just thanking the members of our lab. And this is a photo from our lab, and just thanking you for your time and taking any more questions. This is the escape room in Ann Arbor. Great. So
Any further questions?
I just want to ask you one
Yes.
more thing. So what are your favorite use cases for the unsupervised clustering, for the unsupervised method? For example, we work with organoids. It's not always clear exactly what kind of cells these things are turning into. Are those kind of things that what you're imagining are diseased tissues, like people who have abnormalities? Is that what you think the best cases are, or some other ones I haven't thought of?
exactly. Yeah, I think you just said that. That's pretty good for the use cases. Is that here I just showed one example of we validated a data set that's pretty easy, the cerebellum, but we also know a lot about it. But I think organoids is a really good use case. Another thing that comes up is abnormalities that are specific to an individual, such as cancer. So your particular cancer might not actually match really well to a reference with the supervised methods. And a lot of times people say, hey, we can label every cell type in this data set, but we can't label the cancer cells. Well, you can actually use a mixture of supervised methods for the known ones and unsupervised methods for the cell types that you can't label. So I would say whenever you have some kind of specific disease that has maybe a lot of variance that's not captured with a really well-known reference, or you have a lot of these platform effects that I talked about, and you might get more accurate just by doing it on the target technology. Great, and it looks like we have a couple more questions on Zoom here, and then I'll try to wrap up soon because I know we're at time. Cross-platform issues. Some cell types are almost always found in close spatial proximity to each other, underpowered due to multi-collinearity. Yeah, that's certainly true. So the way that our platform effect normalization works is it just combines everything to a pseudo bulk. So the way we do it is we don't take the spatial location into account, so everything just gets merged together because we're looking for specific effects. That's just how we do it. It's not the only approach. And then have you tested these methods with Xenium data or FlexSeq? So Xenium is similar to Merfish. It's going to be imaging based. Actually, we've tested it on those, but I didn't show that today here. And then FlexSeq as a reference. I'm not familiar with that specific case, so yeah, if that's all, then I just want to thank everyone for their time and happy to discuss. You can also contact me at my email here, dmcable@umich.edu. So thank you.
The whole talk on cell identification and statistical methodology without the term AI. That'd be amazing. Thank you. One.
Thanks for inviting.
Yeah, we get a lot of